Chapter number 12, Mineral Nutrition. The basic needs of living organisms are essentially the same. They require macromolecules such as carbohydrate, protein, and fats, and water and minerals for their growth and development. This chapter focuses mainly on inorganic plant nutrition, wherein you will study the methods to identify elements essential to growth and development of plants and the criteria for establishing the essentiality. You will also study the role of essential elements, their major deficiency symptoms, and mechanism of absorption of this essential element. This chapter also introduces you briefly to the significance and mechanism of biological nitrogen fixation. Method to study the mineral requirement of plants. In 1860, Julius von Sech, a prominent German botanist, demonstrated for the first time the plant could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique of growing plants in a nutrient solution is called as hydroponic. Since then, a number of improvised methods have been employed to try and determine the mineral nutrients essential for plant. The essence of all these methods involve the culture of plant in a soil-free defined mineral solution. This method requires purified water and mineral nutrient salts. Can you explain why this is so essential? After a series of experiments in which roots of the plant were immersed in nutrient solution and wherein an element was added, removed, or given in varied concentration, a mineral solution is suitable for the plant growth has been obtained. By this method, essential elements were identified and deficiency symptoms were discovered. Hydroponic has been successfully employed as a technique for commercial production of vegetables such as tomato, seedless cucumber, and lettuce. It must be emphasized that the nutrient solution must be adequately aerated to obtain the optimum growth. What would happen if solution were poorly aerated? Diagrammatic views of the hydroponic technique is given in figures. Essential mineral elements. Most of the mineral present in soil can enter plants through roots. In fact, more than 60 elements of the 105 discovered so far are found in different plants. Some plant species accumulate selenium, some other gold, while some plants growing near nuclear test sites take up radioactive strontium. There are techniques that are able to detect the mineral even at a very low concentration, that is 10⁸ gram per milliliter. The question is whether all the diverse mineral elements present in a plant, for example gold and selenium as mentioned above, are really necessary for plants. How do we decide that it is essential or not for the plant? And criteria for essentiality. The criteria for essentiality of an element are given below. The first, the element must be absolutely necessary for supporting normal growth and reproduction. In the absence of that element, plants do not complete their life cycle or set the seeds. Second, the requirement of the element must be specific and not replaceable by another element. In other words, the efficiency of any one element cannot be met by supplying some other element. Third, the element must be directly involved in the metabolism of that plant. Based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on their quantitative requirements. First is micronutrients and second is micronutrients. First, macronutrient. Macronutrients are generally present in plant tissues in a large amount in excess of 10 millimole per kilo of dry matter. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, magnesium. Of this, carbon, hydrogen and oxygen are mainly obtained from CO2 and H2O, while others are absorbed from the soil as mineral nutrient. Micronutrients or trace elements are needed in very small amounts, less than 10 millimole per kilo of dry mass. This includes iron, manganese, copper, molybdenum, zinc, boron, chlorine, and nickel. In addition to the 17 essential elements named above, there are some beneficial elements such as sodium, silicon, cobalt, and selenium. They are required by higher plants. Essential elements can also be grouped into the four broad categories on the basis of their diverse function. These categories are essential elements as components of biomolecules and hence structural elements of the cells. Example, carbon, hydrogen, oxygen, and nitrogen. Essential elements that are components of energy-related chemical compounds in plants. Example, magnesium in chlorophyll and phosphorus in ATP. Essential elements that activate or inhibit enzymes. For example, magnesium is an activator for both ribulose, biphosphate carboxylase, oxygenase, and phosphophenol pyruvate carboxylase, both of which are critical enzymes in photosynthetic carbon fixation. Zinc is an activator of alcohol dehydrogenase and molybdenum of nitrogenase during nitrogen metabolism. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathways you have studied earlier. Some essential elements can alter the osmotic potential of cell. Potassium plays an important role in the opening and closing of stomata. You may recall the role of minerals as solutes in determining the water potential of a soil. Role of macro and micronutrients. Essential elements perform several functions. They participate in various metabolic processes in the plant cells, such as permeability of cell membrane, maintenance of osmotic concentration of cell sap, electron transport system, buffering action, enzymatic activity, and act as major constituent of macromolecules and coenzyme. Various forms and functions of mineral elements are given below. Nitrogen. This is a mineral element required by plants in the greatest amount. It is absorbed mainly as NO3, though some are also taken up as NO2 or NH4. Nitrogen is required by all parts of the plant, particularly the meristematic tissues and the metabolically active cells. Nitrogen is one of the major constituents of proteins, nucleic acids, vitamins, and hormones. Phosphorus. Phosphorus is absorbed by the plants from the soil in the form of phosphate ions, either as H2PO4 or HPO42. Phosphorus is constituent of cell membranes, certain proteins, all nucleic acids, and nucleotides, and is required for all phosphorylation reactions. Potassium. It is absorbed as potassium ion K+ in plants. This is required in more abundant quantities in the meristematic tissues, buds, leaves, and root tips. Potassium helps to maintain an anion and cation balance in cells and is involved in protein synthesis, opening and closing of stomata, activation of enzyme, and in the maintenance of turgidity of cells. Calcium. Plant absorbs calcium from the soil in the form of calcium ions Ca2+. Calcium is required by meristematic and differentiating tissues. During cell division, it is used in the synthesis of cell wall, particularly as calcium pectate in the middle lamella. It is also needed during the formation of mitotic spindle. It accumulates in older leaves. It is involved in the normal functioning of the cell membranes. It activates certain enzymes and plays an important role in regulating metabolic activities. Magnesium. It is absorbed by plants in the form of divalent Mg2+. It activates the enzyme of respiration, photosynthesis, and are involved in the synthesis of DNA and RNA. Magnesium is a constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Sulfur. Plant obtains sulfur in the form of sulfate. Sulfur is present in two amino acids, cysteine and methionine, and is the main constituent of several coenzymes, vitamins, thiamine, biotin, coenzyme A, and ferredoxin. Iron. Plants obtain iron in the form of ferric iron. It is required in larger amount in comparison to other micronutrients. It is an important constituent of protein involved in the transfer of electron like ferredoxin and cytochromes. It is reversibly oxidized from Fe2+ to Fe3+. During electron transfer, it activates catalase enzyme and is essential for the formation of chlorophyll. Manganese. It is absorbed in the form of magnesious ion Mn2+. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is in splitting of water to liberate oxygen during photosynthesis. Zinc. Plant obtains zinc as Zn+2 ion. It activates various enzymes, especially carboxylase. It is also needed in the synthesis of oxygen. Copper. It is absorbed as cupric ions Cu2+. It is essential for the overall metabolism in plants. Like iron, it is associated with certain enzymes involved in redox reaction and is reversibly oxidized from Cu plus to Cu2+. Boron, it is absorbed as BO33 or B4O7. Boron is required for uptake and utilization of Ca2+. That is calcium ion, membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Molybdenum, plant obtain it in the form of molybdenate ion, MoO2. It is a component of several enzymes including nitrogenase and nitrate reductase, both of which participate in nitrogen metabolism. Chlorine, it is absorbed in the form of chloride ion. Anion, Cl- along with Na plus and K plus. It helps in determining the solute concentration and the anion-cation balance in the cell. It is essential for the water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. Deficiency symptoms of essential elements. Whenever the supply of an essential element becomes limited, plant growth is retarded. The concentration of the essential element below which plant growth is retarded is termed as critical concentration. The element is said to be deficient when present below the critical concentration. Since each element, one or more specific structural or functional role in plants, in the absence of any particular elements, plants show certain morphological changes. These morphological changes are indicative of certain elements deficiency and are called as deficiency symptoms. The deficiency symptoms vary from one element to another element, and they disappear when the deficient mineral nutrient is provided to the plant. However, if deprivation continues, it may eventually lead to the death of the plant. The parts of the plant that show the deficiency symptom also depend on the mobility of the element in the plant. For elements that are actively mobilized within the plant and exported to young developing tissues, the deficiency symptoms tend to appear first in the older tissue. For example, the deficiency symptom of nitrogen, potassium, and magnesium are visible first in the senescent leaves. In the older leaves, biomolecules containing these elements are broken down, making these elements available for mobilizing to younger leaves. The deficiency symptoms tend to appear first in the young tissues whenever the elements are relatively immobile and are not transported out of the mature organ. For example, elements like sulfur and calcium are part of structural component of the cell and hence are not easily released. This aspect of mineral nutrition of plant is of great significance and importance to agriculture and horticulture. The kind of deficiency symptoms shown in plants include chlorosis, necrosis, stunted plant growth, premature fall of leaves and buds, and inhibition of cell division. Chlorosis is the loss of chlorophyll, leading to yellowing of leaves. This symptom is caused by deficiency of element nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc, and molybdenum. Likewise, necrosis or death of tissue, particularly leaf tissue, is due to deficiency of calcium, magnesium, copper, potassium. Lack or low level of nitrogen, potassium, sulfur, molybdenum causes an inhibition of cell division. Some elements like nitrogen, sulfur, molybdenum delay flowering or if their concentration in plant is low. You can see from the above that deficiency of any element can cause multiple symptoms and that the same symptoms can be caused by deficiency of one or several different elements. Hence, to identify the deficient element, one has to study all the symptoms developed in all the various parts of the plant and compare them with the available standard table. We must also be aware that different plants also respond differently to the deficiency of the same element. Toxicity of micronutrients. The requirement of micronutrient is always in low amounts, while their moderate decrease causes the deficiency symptom and a moderate increase causes toxicity. In other words, there is a narrow range of concentration which the elements are optimal. Any mineral iron concentration in tissues that reduces the dry weight of tissue by about 10% is considered toxic. Such critical concentration vary widely among different micronutrients. The toxicity symptoms are difficult to identify. Toxicity level for any element also vary for different plant. Many times excess of an element may inhibit the uptake of another element. For example, the prominent symptom of the manganese toxicity is the appearance of brown spot surrounded by chlorotic veins. It is important to know that manganese compete with iron and magnesium for uptake and with magnesium for binding with enzyme. Manganese also inhibit calcium translocation in shoot apex. Therefore, excess of manganese may in fact induce deficiency of iron, magnesium and calcium. Thus, what appears as symptoms of manganese toxicity may actually be the deficiency symptoms of iron, magnesium, and calcium. Can this knowledge be of some importance to a farmer, a gardener, or even for you in your kitchen and garden? Mechanism of absorption of elements. Much of the studies on mechanism of absorption of elements by plant has been carried out in isolated cells, tissues, and organs. These studies reveal that the process of absorption can be demarcated into two main phases. In the first phase, an essential rapid uptake of iron into the free space of or outer space of cells. The apoplast is passive. In the second phase of uptake, the ions are taken in slowly into the inner space, the symplast of the cells. The passive movement of ions into the apoplast usually occur through ion canals, the transmembrane proteins, and functions as selective pores. On the other hand, the entry or exit of ions to and from the symplast requires the expenditure of metabolic energy, which is an active process. The movement of ions is usually called flux. The inward movement into the cell is in flux and the outward movement is called as efflux. You have read the aspects of mineral nutrient uptake and translocation in plants in chapter 11. Translocation of solutes. Mineral salts are translocated through xylem along with the ascending stream of water which is pulled up through the plant by transpirational pull. Analysis of xylem sap shows the presence of mineral salts in it. Use of radioisotopes of mineral element also substantiate the view that they are transported through the xylem. You have already discussed the movement of water in xylem in chapter 11. Soil as reservoir of essential elements. Majority of the nutrients that are essential for the growth and development of plants become available to the roots due to weathering and breakdown of rocks. This process enrich the soil with dissolved ions and inorganic salts. Since they are derived from the rock minerals, their role in the plant nutrient is referred to the mineral nutrition. Soil consists of a wide variety of substances. Soil not only supplies mineral but also harbors nitrogen-fixing bacteria, other microbes, hold water, supplies air to the root and acts as a matrix that stabilizes the plant. Since deficiency of essential minerals affect the crop yield, there is often a need for supplying them through fertilizers, both macronutrients NP, K, S, etc. and micronutrient Cu, Zn, Fe, Mn, etc. form components of fertilizer and are applied as per need. Metabolism of nitrogen. Nitrogen cycle. Apart from carbon, hydrogen and oxygen, nitrogen is the most prevalent element in the living organism. Nitrogen is a constituent of amino acid, protein, hormones, chlorophylls and many of the vitamins. Plants compete with microbes for the limited nitrogen that is available in the soil. Thus, nitrogen is the limiting nutrient for both natural and agricultural ecosystems. Nitrogen exists as two nitrogen atoms joined by a very strong triple covalent bond. The process of conversion of nitrogen to ammonia is termed as nitrogen fixation. In nature, lightning and ultraviolet radiation provide enough energy to convert nitrogen to nitrogen oxide, NO, NO2, N2O. Industrial combustions, forest fires, automobile exhausts, and power generating stations are also sources of atmospheric nitrogen oxides. Decomposition of organic nitrogen to dead plants and animal into ammonia is called as ammonification. Some of this ammonia volatilizes and re-enters the atmosphere, but most of it is converted into nitrite by soil bacteria in following step. 2NH3 plus 3O2 gives 2NO2 plus 2H plus 2H2O. 2NO2 plus O2 gives 2NO3. Ammonia is first oxidized to nitrite by bacteria Nitrosomonas and or Nitrococcus. The nitrite is further oxidized to nitrite with the help of bacterium Nitrobacter. These steps are called as nitrification. This nitrifying bacteria are called as chemoautotrophs. The nitrate thus formed is absorbed by plants and is transported to the leaves. In leaves, it is reduced to form ammonia that is finally forms the amine group of amino acids. Nitrate present in the soil is also reduced to nitrogen by the process of denitrification. The denitrification is carried by bacteria, Pseudomonas and Thiobacillus. Biological nitrogen fixation. Very few living organisms can utilize the nitrogen in the form of N2 available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organism is called as biological nitrogen fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes. Such microbes are called as N2 fixers, where nitrogen is converted into ammonia with the help of nitrogenase. The nitrogen fixing microbes could be free living or symbiotic. Example of free living nitrogen fixing and aerobic microbes are Azotobacter and Bejornikia, while Rhodospirillum is anaerobic and Bacillus free living. In addition, a number of cyanobacteria such as Anabaena and Nostoc are also free living nitrogen fixers. Symbiotic biological nitrogen fixation. Several types of symbiotic biological nitrogen fixing association are known. The most prominent among them is the legume bacteria relationship. Species of rod-shaped rhizobium has such relationship with the roots of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. The most common association on root is as nodules. These nodules are small outgrowth on the root. The microbe Frankia also produce nitrogen fixing nodules on the roots of non-leguminous plants like Ulmus. Both rhizobium and Frankia are free living in soil but as symbiotes and fix atmospheric nitrogen. Uproot any one plant of common pulse just before flowering. You will see near spherical outgrowths on the root. These are nodules. If you cut through them, you will notice that the central portion is red or pink. What makes the nodules pink? This is due to the presence of leguminous hemoglobin or leg hemoglobin. Nodule formation. Nodule formation involves a sequence of multiple interactions between rhizobium and roots of the host plant. Principal stages in the nodule formation are summarized as follows. Rhizobia multiply and colonize the surrounding of roots and get attached to epidermal and root hair cells. The root hair curls and bacteria invade the root hair. An infection thread is produced carrying the bacteria into the cortex of the root, where they initiate the nodule formation in the cortex of root. Then the bacteria are released from the thread into the cells, which leads to the differentiation of specialized nitrogen fixing cells. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrients. These events are depicted in figure 12.4. The nodule containing all the necessary biochemical components such as the enzyme nitrogenase and like hemoglobin. The enzyme nitrogenase is a molybdenum iron protein and catalyzes the conversion of atmospheric nitrogen to ammonia, the first stable product of nitrogen fixation. The reaction is as follows: N2 plus 8 electron plus 8 hydrogen atom plus 16 ATP gives 2NH3 plus H2 plus 16 ADP plus 16 P. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic condition. The nodules have adaptation that ensure that the enzyme is protected from oxygen. To protect this enzyme, the nodule contains an oxygen scavenger called as like hemoglobin. It is interesting to note that this microbe lives as aerobes under free living condition where nitrogenase is not operational, but during nitrogen fixing events they become anaerobic, thus protecting the nitrogenase enzyme. You must have noticed in the above reaction that the ammonia synthesized by nitrogenase requires a very high input of energy, 8 ATP, for each NH3 produced. The energy required thus is obtained from the respiration of the host cell. Fate of ammonia. At physiological level, pH, the ammonia is protonated to form NH4 plus ion. While most of the plant can assimilate nitrate as well as ammonium ions, the latter is quite toxic to plants and hence cannot accumulate in them. Let us now see how NH4 plus is used to synthesize amino acids in plants. There are two main ways in which this can take place. First, reductive animation. In this process, ammonia reacts with alpha-ketoglutaric acid and forms glutamic acid as indicated in the equation given below. Alpha-ketoglutaric acid plus NH4 plus NADPH, and in the presence of glutamate dehydrogenase gives glutamate plus H2O plus NADP. Transamination, second equation. It involves the transfer of amino group from one amino acid to the keto group of keto acid. Glutamic acid is the main amino acid from which the transfer of NH2, the amino acid, takes place, and another amino acid are formed through transamination. The enzyme transaminase catalyzes all such reactions. For example, the equation is given below. The most important amides, asparagine and glutamine, found in plants are structural part of proteins. They are formed from two amino acids, namely aspartic acid and glutamic acid, respectively, by addition of another amino group to each. The hydroxyl part of acid is replaced by another NH2 radical. Since amides contain more nitrogen than the amino acids, they are transported to the other part of the plant via xylem vessels. In addition, along with the transpiration stream, the nodules of some plants, example soybean, export the fixed nitrogen as urides. These compounds also have a particularly high nitrogen to